Hello, I'm VB and I'm coming to you live from Pioneer Brewing in Orange, New South Wales, Australia. Today I'm going to be talking about craft beer micro on a budget. I'm here currently consulting and assisting with Pioneer setting up our laboratory, which is quite exciting. And today we'll be talking through lactic acid bacteria. So we'll be talking about the lab, a space to where you want to prepare and review microbiology. We want to be looking at HLP media, how to prep a sample, how to review the growth that we find, and what to do with the results, and then looking at possible locations in the brewery where problems can occur. So the very beginning is to have a dedicated space that's limited with airflow that you can have your brewery lab to be. So this might be in a kitchen, this might be just an old cupboard somewhere, or a converted shipping container, which is what we're doing here at Pioneer. When we look at basic equipment, you want a microscope to be able to identify what's growing in your media. You can either cook media up via a stir plate or a pressure cooker, like a demo that I'll do a little bit later. Definitely need some scales that are 0.01 grams so that you can weigh out media accurately. And then you want to put that media with water in borosilicate glass. So you don't want to be using just any glass, you want borosilicate so that it can handle being autoclaved. Then you need somewhere to incubate your samples that is warm. So that could be next to the boiler, your hot liquor tank. It might be something as a heat mat from a homebrew store in an esky with some kind of thermometer that cuts in, cuts out. And there's different methods for this. And micro equipment doesn't have to be expensive. Then you're going to have sample ports. So that's where you're going to take your sample. So these could be standard sample ports on tanks, such as Zickel sample ports or your other standard sample ports. The main thing is that you want to have those pulled down and clean during your CIP. And you also want to make sure that these sample ports are flamed with ethanol so that they are sanitary when you're taking the sample. When you talk about more detailed sample ports, that will be the aseptic sample ports. So basically the aseptic sample ports will usually be filled with ethanol, and that's when they're closed so that they're sanitary. And then when you open those sample ports, the ethanol will come out and the product will come through. Now there are also aseptic needle ports where you've got to jab through a membrane, but those membranes get about 18 jabs before they're replaced. So the preference is definitely on the aseptic sample port, but they're a bit more expensive. Then again, you need to clean these and make sure you have adequate flow. So your more expensive aseptic sample ports will have a CIP clip that can be part of your loop. If your sample ports are down on the base of the tank, you know that you're going to get adequate flow through it. However, if it's protruding out further up the tank, you want to take that sample port off and put it as part of your CIP loop so that you're cleaning in place. You could share a sample port, so you could have a valve behind it and double valve, and it could live in a sterilant bucket, and then you can fill it up with ethanol and flame it. These sample ports here are about 119 USD. So you could just have one if you're on more of a budget and just share it between tank to tank. The main thing is making sure that you always fill the ethanol back up and keep them clean. So the media that I'm going to be discussing today, and there's a lot of different medias out there for lactic acid bacteria. There's things like rack-array. But we're going to be talking about HLP because I find HLP the easiest. And when I started out in the micro lab quite a few years ago, HLP was where we started, and I just found it very, very easy to use. So we're basically going to be looking for LACTOBACILLI species and PEDIOCOCCUS species. And you can see there in those tubes, you've got different growth depending on what the species is. When we talk about the cost, HLP, if you do a bit of a breakdown here, and this is in Australian dollars, with your pipette tips, your gloves and the rest of it, it's only about 13 cents a test. So that's very, very cheap compared to something like BrewPower, which is $43. The difference is going to be the time. So something like the BrewPower PCR is going to give you a result in 3 hours, where the traditional media is going to be a bit slower because the organism has to grow in that medium. When we start out, we need to make sure that we have sterile sample taking. So that means you need a clean work area that you're able to wipe down and sanitize, and you want to have a flame if you don't have a laminar flow. So what's going to happen is that flame is going to sterilize the air by burning with a convection current. And that's basically creating your sterile work area. So on your lab bench, that sterile area would be approximately 30 to 50 centimeters in radius. And that's going to be our sterile work area that we want to work under during our micro testing. Another thing to note when you're taking a sample in a tank, you really want to take a sample where the population of the tank is towards the tail end of fermentation, so you're getting a good mix in that tank. And also, you know, most of the yeast is consuming the sugars and the rest of it, so other things are starting, if they are there, are starting to consume the other metabolites of the tank. This could also be when you transfer a tank, when you're carbonating a tank, when you're chilling a tank, you're getting those convection mixing, or when you've transferred or say filtered through a centrifuge, you're getting a good mix. Tanks that are on chill for a long time would be considered a layered sample, and you might be getting the entire population of that tank. When we come onto the micro floor, I've always used flame, and I've always had someone else use the flame and follow the safety requirements there because it is a bit dangerous. I have seen in some newer breweries the use of steam. The reason why is you're going to spend all this time making the sample. You want to make sure that that sample is aseptic. So it's the same principle as setting up the flame in the micro room. So what we do is we let the ethanol out, because obviously that's a flame risk. You then lightly burn the end of the aseptic sample port. You then get someone to safely hold that flame, and you want to feel that it's nice and warm, but it's not going to burn you. And then you want to let a bit of that sample of the beer out to cool that sample port, because we don't want to pasteurize the sample that you're going to test. And then you want to fill that sample up, and that's what we'll take up to the micro lab. Prepping out HLP is pretty easy. If you're going to make up 100 mil, and I'll show you in a little bit, we're going to just make up 50. You want to add about 7 grams of HLP, and you want to add usually a little bit of agar as well, is my recommendation. Now, there is cyclohexamide in this medium, and it will affect you just like it affects yeast by stopping that budding. So you really want to make sure that you have a P2 mask on when you're using this agar and prepping that. And then we just weigh out the required amount. Now HLP only needs to boil. So you can do this with a stir plate with that heats, and once it boils you can just stop. You can use a pressure cooker, which is what I'm going to demo today. I wouldn't recommend using a microwave, because if you overcook the cyclohexamide that's going to prevent our yeast from growing in that media, then you could actually, you know, affect the cyclohexamide. So I wouldn't recommend a microwave, and I don't think it's very safe to use one. So when we're starting out, first with micro in a brewery before that hasn't tested, we want to start at about 0.1 mil. Then look at those results, and then move up to 1 mil, and then when you're more confident you can move up further. And then you can top up with about 10 mil to 15 mil of HLP. So you want to get that sample, put that 1 mil in a centrifuge tube, and then pour in your HLP agar. Then invert it. And I'll show that a little bit later. With any method in a laboratory, I always suggest that you have a positive and negative control. So in this example here, the positive is control. We've used a little bit of sour pitch because we know that's Lactobacillus plantarum. Then we have our sample of beer, and we have a negative control. Now the negative control is just the agar itself to make sure that the agar was boiled enough to become sanitary. Then we want to incubate this in our incubator to see if something grows. Now, if there is a serious infection with something like Lactobacillus brevis, for example, you'd probably have a result within 2-3 days. When you're using the sour pitch, that grows quite fast as well. That's one of the beneficial reasons that culture was selected for Lalanon to use in sour beers. Things that are going to be small, slower growing are things like Lactobacillus linderi, and PEDIOCOCCUS could even take up to 12 days or more, maybe even up to 14, depending on how much of a problem you have. Now I'm just going to get ready to set up this demo of preparing the media. Okay. So what I'm going to do here is we're going to have some scales, which I'm just setting up. I'm going to put on some PPE, so I'm going to put on some gloves. I'm going to put on my P2 mask. And we're just going to get out our HLP media, a weigh boat. So we'll turn our scales on. We will tare our scales. And I'm going to be weighing out about 3.5 grams of the HLP media because I'm going to be putting it in 50 ml of water, of distilled water. Okay. Now I've pre-prepared my distilled water in here. And I'm going to gently pour in the HLP agar. Going to gently swirl that. Now, what you want to make sure is that you don't fully tighten the reagent bottle. You want to about a quarter crack so that the lid will hold on, but you're not creating a pressure where you're not going to be able to get that off. So just lightly cracked. Then we want to cook that media. So here I have a pressure cooker. I'm going to add about 600 ml to this pressure cooker. I'm going to put in my HLP, seal the pressure cooker, and I'm going to put this on steam setting is what you want, and I'm going to put it on for 21 minutes. And I'm going to start that. So while that is cooking, I'm just going to go through a few possible locations in the brewery that we could be having an issue. So these are some of the tips from my experience in my previous brewing career, but also visiting a lot of breweries around Asia Pacific at my role at Lallemand, and also bits and pieces picked up from colleagues at Lallemand as well. So if we look at terms of the result, what happens is once these agars have grown, there's going to be some colonies in there if there is an issue. Now, typically, things like Lactobacillus brevis are going to look more like teardrop colonies. Pediococcus damnosus will look more like raindrops, comment likes. Some wild yeast media can grow inside HLP, so it's another reason to have a look under the microscope. And sometimes if you have a high acidic bacteria contamination, then it will cloud the media and you might need to dilute. So basically, how many cells that you count here equals a colony forming unit, and that'll be how many cells of the sample that you took. So say if it was a mil, how many cells of a problem that I have within my sample of beer, and that equals the colony forming unit. Now, sensitivity, like I said, you can start out at 0.1, then move up to a mil, 50 mil, 100 mil. This is all based on your house specification. So you need to set up a spec of what are you going to accept for your internal house specification of what a problem is. You know, if it's greater cells. For us in my previous micro role, it was 0. Now, how far do you want to go with a result? So basically, something has grown. You open up the tube and have a sniff. Does it smell sour? Do we put it under the microscope and have a look? Some breweries that I set them up with a small micro lab using HLP are happy enough if there's a result, then they will change their CIP if there's any result, because at the end of the day, if something's growing in there, it shouldn't be there in the first place, so they just change it. Other people will put it under the microscope and have a look, maybe take some photos and send that off to some other colleagues in larger brewing labs. There's a great book there on the colour beverage of biology that has pretty much a whole bunch of agars and different species you can use for identification. You might send the sample off if it's a repeat issue to have it properly identified, over to another lab to get some genetics done to work out exactly what it is you're dealing with, to then work out how to eradicate it. So here are some example results. So say if you have 2 cells of Lactobacillus brevis, in 5 days, in 1 mil, in that 5 days it'll cause sourness and turbidity. Pediococci, first 4 cells per 1 mil of beer, about 14 days until you get diacetyl and you start having other issues there. Now keep in note that some Lactobacilli and Pediococci species are inhibited, and that's one of the reasons that Lallemand, as well as the flavour and souring time of sour pitch, or how Verticus pitch these were chosen, is that they can actually be inhibited by hops. So a lot of the time it's not actually going to spoil the beer in the presence of hops, depending on the alpha and beta acid. There's also a genetic test for this where you look for the HOR-A and HOR-C genes to see if those bacteria are hop resistant, and if they are resistant to the hops, then most likely they will spoil the beer. So can we save it? It's really up to the brewery at the end of the day, whether you want to do pasteurization, whether you want to dump the beer, whether you want to use sterile filtration. It's more recommended to use a 0.65 if it's a really hoppy beer. You know, you could dry hop it, add a bit of cinema to change the colour and call it a late hopped sour. The big thing is to sanitize everything. So even when you're putting together pipe work, you want to be spraying sanitizer like ethanol over those triclovers putting together the pipe work. If you think about it, it's getting hot and it's getting cold as part of those CIPs. And so if there's a little bit of, say, brevis on a triclover is sitting just enough between it that when it's hot, it's withstanding the heat and not really getting in with the sterilant. And then you cool that down with your peracetic acid and your wort, and that particular organism could enter into your pipework even though you've done a CIP. So spraying everything is definitely highly recommended. Another area to look for is the heat exchanger. So eventually you will need to pull apart your heat exchanger, but definitely doing different acid mixes, forward and back flushes, caustics, leaving phosphoric nitric in it over the weekend, for example, is another way to just regularly change the CIP that you're doing on the heat exchanger. Now, if you are using things like phosphoric nitric, you want to make sure that there's not chlorine in your brewing liquor, as this will react with the acid and really degrade the seals within the heat exchanger. Also, if you're going to the point of pulling apart your heat exchanger, you want to make sure that you label it, as sometimes they're slightly bent and putting it back together the wrong way, they won't seal. A really simple test for the heat exchanger is what we call the pop top test, or you can use the little sample containers. Now, basically what you want to do is just after the heat exchanger where it's cooled down, you want to have a sample port, and you can put a sample under flame in one of those little yellow containers or the pop top container, and you can just put that in the agar incubator. And if that pops or it gets turbid or it looks like it's bubbling away, then you know something is wrong with the heat exchanger because you haven't added yeast yet to your beer. So this is a really simple method, and it can save you a bit of extra plating and agar time, because if the wort's integral in the first place, then you might not need to plate it every single time. Once you discover that there is some type of fermentation in this samples, then what you'll want to do is then probably start plating to kind of identify what it is, or you might change your CIP regime on that heat exchanger. Spray balls. So spray balls sometimes can have things stuck in them, like little bits of triclover seals or other things that prevent them from spinning. So this will leave blind spots in the tank. Now, in the early days of the previous brewery that I used to work in, we had a theory that there was an issue with the spray ball in the tank, and we added basically dry extract powder and put that around the door at the top of the tank and also on the dry hop port, and then because it's very sticky, and we ran just hot liquor to see if it would actually clean the tank, and what we found out is that there was shadowing because that spray ball was not long enough and it wasn't effectively cleaning the whole tank, and that was where the lactobacillus at the time was living inside the fermenter. And so if the fermenter went a little bit over, or you overfilled the tank a bit, or the krausen of the yeast went up, then that's how it was infecting the beer. So that's a real simple, easy tip to check that your spray balls are definitely cleaning that tank if you don't have an ATP meter. So then you also want to look at testing your CIP regime. So the easiest simple test out there for $5 a test is an ATP test. So that's going to look for bioluminescence and see if anything is alive. You can't use that in your chemicals, but you can do it in your rinse water. It works on the Firefly enzyme giving off ATP, and then you get a light result. So it might have a pass, fail, or warn. If you have that, then you want to re-clean that tank. Now, keep in mind that if your hot liquor is not hot enough, you will still have some positives on ATP, because it's obviously not specific, it's looking for any organism to give off ATP. So if your hot liquor is not clean enough, you might get a result from just the hot liquor itself, and the tank itself might actually be clean, but you might need to heat that hot liquor up a bit more, or have some better filtration in place. There's also 3M Petri film out there. That's another method of just putting a mill on, squeezing down, and it gives you a result in a couple of days for different types of bacteriums. The other big one is titration for your chemicals. It's really, really simple. So you know that you have a known volume that you put your liquor into the tank, and say that that comes out at the racking port is where the water comes out, so you have a known volume. If you don't have a flow meter, you then add your litres of caustic, and then you're aiming at about 3%. Now, how do you actually know that it is 3%? You can take some of that caustic out. You can get a simple titration kit from your chemical supplier. Basically, you put one chemical in, you count how many drops that you put in, and when that chemical reaction, in this case is pink, turns to clear, that number of drops with a simple occasion can tell you what level your caustic is. So it might be that you're thinking you're dosing at 3, but you're only dosing at 1, or you might be dosing higher, and you can actually save some money there on caustic. And each of your chemicals that you use in the brewery, your chemical supplier should be able to find and supply a titration kit. So it's a really, really simple test. It's very similar to fish tank pH testing. Now, another thing is how is the microflora of your brewery, and every microflora of the brewery is going to be different. How is it entering, and what's happening? So you need to think about a bit more about return growlers and bottles and kegs. If you're using rental kegs, you know, they might have a Brett beer in there before, or a sour or something else, and now you're cleaning that keg within your brewery, so you're bringing those organisms in. Grain, we'll go through a little bit when we set up the positive control, is a good indicator that you really want to make sure you're cleaning up that dust, the spent grain you're getting out of the brewery. You're not leaving it there to come in contact with air and blow around the brewery. Also shoes, food, equipment, everything else that's brought in, and just leaving things dirty, like in this example, the breather bucket on the floor that hasn't been properly cleaned out, or gunk built up because you've used it off the tank that day, but you haven't properly hosed that out. So that's all stuff that's going to dry and build up and become a problem later. Floors and equipment. You want to make sure that you don't have dirty floors. You really should be rinsing those floors down and getting any dirty equipment out of the way. If you have porous concrete, you can do some foam downs. I really do like the tiles that you see in some of those European breweries. Nice and clean and great draining. Another great cleaning chemical and regime is to foam down. So that's using something like CCF, and basically you foam all of the tanks, and then you want to make sure that you rinse all of that off and rinse all that away with this drain. Just because you've used that chemical to clean down all of the equipment, once you have killed those organisms, those pools of water, as the chemical breaks down, are going to be nutrient sources used by other bacteria. So you want to make sure that once you have foamed down, you thoroughly wash away. Now you can do swab tests of the tanks and things like that, or you could just simply put out some agar plates and then incubate those plates. And you find that when I used to do this at one of the previous breweries I worked for, you know, something like there'd be 200 colonies growing on an agar, and after a foam down, there'd be like 5. So it makes a massive difference. CO2 lines. So you want to make sure that you have a one-way valve, so you're not putting beer back up those CO2 lines. and then you're re-inoculating a problem from one tank into other tanks. So you can set up some kind of custom manifold that will assist in cleaning those lines, or you might just use a clear hose. Once it's been compromised, you'll throw that hose out and clean through a new hose. Another simple way to see if your CO2 lines need cleaning is you can actually, this is a little thing I designed at one of the past breweries that I work for, where we could rig it up to blow CO2 through some already previously autoclaved water. Then we could inoculate that into agar or membrane filter it, just to see if all of our CO2 lines were indeed clean or whether they required a cleaning. So that saved a bit of the CIP time, that maybe you don't need to do it every month. You could do it every 3 months or 6 months, as long as you're doing a simple test like this. The ultrasonic bath, which they have here at Pioneer, that's a photo of it, is a great way to clean not only sinter valves, but also all your parts and equipment. So this ultrasonically cleans by putting sound waves of vibration through the equipment, but this also has caustic and you can heat it. So it gives a very, very vigorous clean, and then those things can be rinsed and put back on the tank as part of the rest of the CIP steps. Okay, so what we might do now is I'll set up to prep our samples. And then we'll try and pour out our agar. Okay, so what I've done here is I've grabbed a sample from the tank, and I have grabbed just some grain dust and a bit of some of the grains, and that's going to be our quick and easy positive control because you're going to have lactobacilli and things living on the grain. Because that's going to be such a high concentration, I've done a serial dilution of 1 in 10 just to bring that down. I've then set up my tubes. So here I have my positive control, and that's going to have the grain dust mix in it. I've got my sample, which is the tank of beer. I have my negative control, and the negative control is just the agar itself to make sure that it has definitely been cleaned. Now, if you look at a result, here is a result from just the grain dust. You can see it's quite heavily populated because we didn't do a serial dilution at that particular one. So there's a lot of acidic as well as lacto and everything growing in this. It's quite a high concentration. So sometimes you will need to do a serial dilution. So I'll just get my work area set up. Okay, so I'm just going to spray down some ethanol here. Just to clean my workspace. We've got my samples here. I'm going to sanitize my gloves and get my flame ready. So there again, we just want to see that is creating a sterile work area, and I can feel that convection current in there. I'm going to get my pipette oil, and I'm going to set it to 1 ml. I'm going to get my pipette tips. And first I'm going to get the sample of the beer. So we're just going to get that ready. And now this sample of beer, if it's a little bit foamy, you might need to just blow it back in with that tip and then pick that back out. I'm then going to put that in. I'll flame the end of that, I'm going to dispose of that tip. And there we can see we've got 1 ml of our sample, and I'm going to fill that up to about the 14-15 mark once our HLP comes out of the pressure cooker. So that's our sample done. Now we want to do our diluted grain dross, and that's going to go into our positive control tube. So again, going to get a fresh pipetta. Now keep in mind when you're pipetting, you've got 2 steps on a pipetta. So the first step is to gently cool down and pick up the solution, and then you use the step to push that out, and then the further step to completely clear that out. Now always be very gentle with a pipetta. Don't go super fast, because what you could do is actually pull, if you don't have a filter in here, you can actually pull your sample up into the pipetta, and then you're spiking all your samples. So we're going to throw this in our positive control. We're going to dispose that tip. And we'll just see how far out it goes away. I'd also recommend that you definitely pre-spray your hands each time you move through that step, so I should have done that there. Okay, that's our HLP. You can see now it is in liquid form. So we would let that cool down, and you basically want to have that at the temperature of baby formula. So you can just lightly tap that against the back of your wrist, and that's going to let you know that it's cool, that the agar has not set, but the agar is ready to pour. So it should take a little bit of time. Sometimes you can put it in a little bit of a water bath to cool down. So the first thing I'm going to do is I'm going to put it in my sample. Now, if it is extremely hot, you're going to pasteurize what you're trying to look for. So you really need to make sure that you cool down and make sure that agar is nice and cool and not going to kill everything that you're trying to look for. So I'm just going to gently pour that in there. Just flame the end of that, flame the end of that, flame that. And then you just want to invert that. So you're mixing that up. And we'll do our positive control. And we want to invert that. Now, negative control, is just going to be the agar itself. And there we have our positive control, our beer sample, and our negative control. And it's really that easy to do micro. And it's not very expensive at all. All you need is a pressure cooker. It's about $80, some tubes, pipetta, and you can do some very simple micro in your brewery and do quite a lot of tests. So what I will do now is just look if there's any questions. On the comments page. Okay. Doesn't look like I can see any questions. I'll just put a test in there in case I can't correctly see. Okay, well, if there is no questions on that, you can contact me by my Lalimond email, which I'll put up now. But thank you for watching today's live, just to show you a bit about lactic acid bacteria testing and possible locations that it could be in the brewery. And it's a very simple, cheap, easy way to look for organisms within the brewery. You can get the HLP media from Dolher now, so it's still under the Siebel brand, but it's joint with Dolher where it's produced. Okay, thanks everyone. I'll just put my email address up and enjoy your day.